Okay, we are picking up where we left off after drawing day 1. So if you bring this drawing back out, we're again dealing with the G protein couple receptor. So you can see G protein was inactive, had GDP attached, ligand binds, causes shape change to the receptor, which allows GTP to now kick out GDP. You have an active G protein who then moves through the membrane, comes into contact with adenylyl cyclase within the plasma membrane, and now we have an active adenylyl cyclase. So shape change for him. When he is active, he's going to continue signal transduction. So this is where we're going to pick up with, is right here. What does that mean, this when he becomes active? All right, so let me move him up this way. So when he becomes active, what does he do? This is going to be an example of what he does. I'm going to send in, you can see I'm going to have to kind of write over this guy. So for step 7, the continuing process, we're going to send in ATP. ATP is going to come in. Adenylyl cyclase will convert ATP into something called cyclic AMP. Cyclic AMP is a second messenger, and this is going to be our first messenger, the ligand here. He's our second messenger, very common second messenger used in cells. He's going to pass the message into the cytoplasm of the cell. So what does he do inside the cell? Well, he's going to activate our kinase. So let me draw kind of a little guy here. He is going to be the guy that activates and takes an inactive protein kinase. I'm going to just number it number 1. Inactive protein kinase number 1, and he's going to turn it into active protein kinase number 1. So cyclic AMP converts inactive protein kinase to an active protein kinase. So this is the first one. So he is our second messenger. So if you need to make a little message here, second messenger. Right? He's in the cytoplasm doing his job. So now you have an active protein kinase number 1. What is his job? Well, his job, remember what kinases do, they phosphorylate molecules. So an active protein kinase number 1 is going to activate, well, we're going to start with our inactive protein kinase number 2, and he's going to activate him into an active protein kinase number 2. Well, in order for him to do that, this is a kinase. What do kinases do? They add phosphates, right? They phosphorylate. So that means in order to do that, we have to send in ATP to do that, right? Kinases use ATP, add a phosphate to this guy, that activates him, turns him into the on position. So yes, phosphorylation in the examples we're talking about activate molecules, like turn them into the active position. Is it possible for a phosphorylation process to inactivate things? It is. That is not how we're talking about them, though, in this example. In G protein couple receptors, the kinases are adding phosphate groups to cause this to become active. So now we have active protein kinase number 2. Well, what is his job? His job is to take an inactive protein kinase number 3, and he is going to turn him into an active protein kinase number 3. So that's what kinases do. In this case, this kinase is going to use an ATP to phosphorylate the inactive kinase 3 and turn him into an active kinase 3. So now we've got active kinase 3. What's his job? You're like, well, we know we're getting to the end here because I'm running out of paper. Right, so active kinase 3, he's going to take some protein that is in the cytoplasm, so inactive protein, and he's going to turn him into an active version of that protein. Right? So there could be some enzyme, not a kinase enzyme, but some other enzyme in the cytoplasm of the cell that needs to be activated. But since this is a kinase, how does he do that? He uses a phosphate group, phosphorylates the inactive protein, and turns him into an active protein. So what's the purpose? Now that why have we activated this protein or this enzyme, for example? This is what we're going to get as our cellular response. All right, so this is that all of this is the signal transduction, passing of the message so that we can ultimately get a cellular response. So for example, in a liver cell, epinephrine or adrenaline binds here, and after all this signal transduction, we're going to take an inactive enzyme, turn them into an active enzyme, and that enzyme will convert glycogen back into glucose. So now that would be the cellular response. So the liver cell is now able to release glucose so that you can now run for your life, as an example. So when we look at this process, like why? Let me back up, zoom out for just a second. Why do we need all of this to get to this? Why can't we just, okay, we've activated him, why can't we just skip down to this guy? Well, there is a reason for doing it this way. So if we come back up here, right, we have one ligand binding to one receptor. So I'm going to show one receptor can activate 100 G proteins. What? That's a lot, right? Let me zoom in here a little bit so it's easier to see. The whole purpose of this whole cascade reaction is amplification. So if one receptor can activate 100 G proteins, like how does it do that? That means attached to this receptor, there's lots of these guys. There's not just one. And so when he shape changes, a whole bunch of these G proteins are going to be able to have GTP kick out the GDP. And so now you have a 100 G proteins now moving through the cell membrane, hitting and coming into contact with adenylyl cyclases. So each one of these will come into contact with one of these. So this is going to be a 1-to-1 ratio. But this one ligand was able to activate 100 of these. So now we're over here. 100 G proteins activate 100 adenylyl cyclases. So this itself, this part is not actually an amplification. This is a 1-to-1, but obviously this is an amplification. But now you've got 100 adenylyl cyclases that are active. Well, now what? Now that he is active, he is now able to activate. He stays active for a while. Remember, every time when the GTP takes a while to hydrolyze, the G protein takes a while to hydrolyze GTP into GDP. And so while he's still stuck here, he's still working. He's still taking ATP to cyclic AMP, ATP to cyclic AMP. So as long as he's still active, he's doing this motion. And because he's able to do that for a while, we end up with one adenylyl, I have to think how to spell this guy, it's kind of strange, cyclase. Adenylyl cyclase can activate 10,000 cyclic AMPs. Right? So we started with one receptor, so all the adenylyl cyclases that were activated end up with now creating 10,000 cyclic AMPs. So our one ligand, we're now at 10,000 cyclic AMPs. That's a huge jump. We went from 1 to 100, but now as we jump on down the pile here, we've now activated lots of cyclic AMP second messengers. And so now these are flooding throughout the cytoplasm. And as they flood throughout the cytoplasm, they are activating inactive protein kinase 1s and turning them into active protein kinase 1s. Now all of these guys are able to move through the cell, and they're going to cause this activation. So we're going to continue to amplify as we go. I'm not going to track all of the numbers in here, but I want you to see that each kinase, like for example, each kinase 1 can activate several kinase 2s. and so on. So if each one can activate more than just, it's not just a one-to-one, like I'm active, I can do several of these, for example. I don't know why I'm so kind of like shoved over to the side. If one of these guys can activate several of these, one of these guys can activate several of these, one of these guys can activate several of these, you're going to get continued amplification as you move down, so that by the time you get to here, you're at this active protein here, you're now at millions. Right? This is amplification. So yes, it may seem kind of annoying. You're like, or confusing at first as to why we have to do this whole domino cycle just to get down to here. But because we don't just have cyclic AMP activate this guy down here, because your cell goes through this process, we have now amplified our signal. So for every one epinephrine molecule, you're going to have millions of these active enzymes, so you're going to get a much bigger response. So hopefully that makes sense. So this whole process, this cascade, leads to amplification. So obviously, you can't just, once this one ligand guy up here binds, it's not like you're just stuck, this liver cell, for example, is stuck going through its cellular response for all time. We have to have a way to shut this down. So we're going to shut this down by making sure the ligand concentration obviously has to fall, right? If you get rid of the ligands, there's going to be enzymes or things in here that get rid of the ligands. So if he's gone, he goes back to his normal shape. He goes back to his normal shape. Remember, this active G protein will eventually hydrolyze GTP to GDP, and he moves back. He's in the inactive state. As long as there's no ligand here, then the message is going to stop from this perspective, right? So that's how you would shut like this piece down. Get rid of the ligand. Have the concentration of ligands decrease. Now down here, how are we going to, I guess I should write that down. To stop, decrease concentration of ligand. So however that decrease of concentration happens, it happens. Like for example, in here, there could be an enzyme in between cells that breaks him down. There may be some kind of pump that like sucks him back up into the cell he came from. So whatever process, obviously we have to have a decrease in concentration, so therefore we shut this part down. So if we shut this part down, how do we shut this part down? We have all these active kinases, right? They don't have anything to do with the ligand anymore. They're just active in the cytoplasm. So we need a way to get these guys to shut down. So if we shut down active kinase number 2, how do we do that? Because notice these are the ones that were phosphorylated. So since these kinases were phosphorylated, we can go, there's going to be a different enzyme that will take the active guy and turn him back into inactive guy, and that is a phosphatase. That's a mouthful. So phosphatase, he is going to inactivate. Okay, so now he has been inactivated by the protein phosphatase. Same thing is going to happen with this guy. So a protein phosphatase inactivates him, and you'll never guess who's going to inactivate this guy down here also. Yes, protein phosphatase. So anytime we have this ATP being added, it has been phosphorylated, like this inactive guy has become active because of phosphorylation. A protein phosphatase can inactivate him and turn him into the off position and shut this system down. You might be wondering what is shutting this system down. It's a different, it could be this guy, but it could be a different enzyme. So we're not, it's going to depend on who exactly are these kinases. So like I have this as kinase 1, but it also could have been kinase A, for example, right? This could be kinase B, kinase C. So I picked 1, 2, 3, but it could have been A, B, C. I just tried to show you them in order. So I don't want to, there is somebody who is going to inactivate him. We are not going to draw him in because we're not going to learn his specific name. You do need to know protein phosphatase. You do need to know that if you don't have any the concentration of ligands falls, and this is going to shut this part down. Protein phosphatases are going to shut this down, and if this is shut down, then the cellular response is shut down. Okay, so if you have, hopefully this makes things a little bit more clear, but if you are still confused, then I'll make a photocopy of this. We can draw all over it. Just come in and see me. Okay, make sure you take a picture of both this guy and, I moved him out of my way, and so now I can't find him. This guy, it doesn't look exactly like this, but this is my little sample. Take a picture of both and submit them to Canvas.